I'm hoping with this lecture to dispel a huge myth that is out there in the medical field, a large part of the medical field. That myth is that cancer is a genetic disease. And I hope you find the evidence I'm going to review as compelling as I do to prove that cancer is a metabolic disease. And when approached that way, it offers a huge opportunity to treat this devastating disease with far less toxic therapies than are currently being utilized in conventional oncology. And I want to get you to view cancer through a different lens. I'm sure all of you realize that it was a few hundred years ago, merely a blink in geological time, that the world was viewed as being flat, and that was accepted dogma. And obviously today it's accepted as a given thing that the world is not flat. The world is round. We've all seen pictures from outer space to prove that it's round. And so this concept, I believe, is as earth-changing as going from a flat earth to a round earth. Now, a major block in the effort to cure cancer has largely been due to the confusion surrounding the origin of this disease. And make no mistake about it, the origin of cancer is far from a settled concept. There is much confusion surrounding the origin of cancer, and there is a total absence of a unifying theory that explains in logical terms where it comes from. That a unifying theory that can integrate the diverse observations that take place with the origination of this disease. And so the failure to clearly define the origin of cancer is largely responsible for the failure to significantly reduce the death rate. In the last, how long has it been since Richard Nixon declared war on cancer? And the billions and billions of dollars that have been spent in pursuit of finding the cure. And so it's interesting to note that until fairly recently, in the medical dictionary, the definition of cancer was a tumor that spread. And so I liked to say in the past that according to the medical dictionary, it's not cancer unless it is. And that is the kind of double talk that has taken place not only in cancer and across the board in medicine in many areas, but that has led us to be chasing our tails in many cases. So what are the hallmarks? Where can we start to talk about what are we calling cancer? So there's 6 principles that I actually took out of Dr. Seyfried's book. And I'll talk more about him and his research later, but one is self-sufficiency in growth signals. It throws out all the rules for all of our normal cells. Insensitivity to growth inhibitory or anti-growth signals. Evasion of programmed cell death, or what we know as apoptosis. Limitless replicative potential, sustained vascularity or angiogenesis, and tissue invasion and metastasis or spread to other distant parts of the body. So that's the general outline of what we're looking at, this growth of tissue that we're calling cancer. And most researchers since the 1970s consider cancer as a type of genetic disease where damaged genes, DNA, underlies the transformation of a normal cell into what we call cancer, a potentially lethal disease that will kill its host, if you will. The somatic mutation theory, which is cancers caused by mutations in DNA, has guided cancer research and drug development for over half a century now. And it's now coming under attack, interestingly enough, because more and more research is starting to prove that it's not real. But there's still billions of dollars in research. The momentum of the medical industry is hard to turn around like the Titanic. I hope to convince you that consideration of cancer as a metabolic disease deserves a lot more attention than it has been given. But what's interesting is there's some of the research I'll review is 30, 40 years old, and it's basically just been ignored. How many of you are familiar with Bruce Lipton and the book he wrote, Biology of Belief? And in that book, he's a PhD. He used to teach at the University of Wisconsin Medical School. And if you watch the video that he gives around the concept of his book, he explains that he quit his faculty position because he was disgusted at the 30, 40-year-old scientific concepts that were being taught to medical students today. And so he quit and wrote this book and has written a number of different books now and is on a mission to teach people the current state of research into cellular metabolism. So as you can see here, there are many current beliefs around. There are many triggers, genetic possibilities, and now I'm talking about metabolic possibilities. And so Bruce Lipton, in his book, Biology of Belief, talks about just where is the brain in the cell. And most medical people will tell you, well, the brain is in the nucleus, it's the DNA. And he says absolutely, utterly, and completely not. If you remove the brain from any organism on this planet, it dies immediately. And there are no exceptions. If you remove the nucleus from a cell, it can live for 2 weeks. The concept of cloning, I'm sure you're familiar with that the nucleus is pulled out of a cell and a different nucleus is inserted, and the cell, when it starts to grow, will carry out the mission of the DNA that's in the present nucleus. And so what Bruce Lipton strongly suggests is that the DNA is the map. And so saying the DNA is the brain is like taking the blueprints for a building, throwing them out on the table and say, okay, build yourself. Obviously a ridiculous concept. You need an architect and you need a contractor or a builder to interpret those blueprints to make the building come to life. Well, the same is true of every one of our cells. The DNA is not the brain. The DNA, up until recently, we had a concept of the DNA that somewhere between 80 and 90% of it was what they call junk DNA. Well, as the saying goes, God don't make no junk. And DNA, all of it, has a purpose. There's no such thing as junk DNA. And we basically just didn't know, so it was referred to as junk. But we know now that what's crucially important is what is allowed through the cell membrane. And think about that word, the membrane. The membrane is actually the brain of the cell because it determines what is allowed to pass into the cell, and it also determines what is allowed to pass out of the cell. And if there is dysfunction in that process, it influences which genes are turned on, which genes are turned off, which genes run amok. And the backup to the cell membrane is the mitochondria, the energy furnaces. And so this is just a picture of mitochondria, the brain, and cell membrane. It's an extremely complex structure. It's not a piece of cellophane. Okay, so according to Thomas Seyfried, who I mentioned earlier, the DNA mutations at the heart of the somatic mutation theory of cancer are downstream from the damage caused to the mitochondria by and externally to the cell membrane, which starts the cascade of the process. In other words, the damaged mitochondria and the cytoplasm cause the DNA damage, not the other way around. So to construct a single metabolic theory of cancer, Seyfried connected the last remaining dots. And we'll get down the line to talk about Otto Warburg. You've all heard of him. And a few other researchers that have come on the scene that are doing some incredible research. And Seyfried tied injured mitochondria with uncontrolled proliferation, which, as you remember, was one of the hallmarks of this disease. Which is the major pathological feature of cancer. It was documented by Virchow. Those of you who have training in medicine and physiology, I'm sure you recognize the name Virchow, more than 100 years ago. In addition to supplying the cell's energy needs, the mitochondria are the energy furnaces, as it were, in our normal cells. The mitochondria are known to regulate many other cellular functions, including programmed cell death or apoptosis, cell division and differentiation, and angiogenesis or development of new blood vessels. To do these functions, the mitochondria carry on a regular conversation with the nucleus. And when they're relaying signals and materials back and forth to the nucleus on a constant basis. Now, I don't know if any of you have ever seen that movie about, I don't even remember the name, but basically their premise for the movie was, you don't ever want to be inside of a cell, because the rate at which things happen, stuff is happening, molecules racing around and substances and organelles, you would quickly want to be somewhere else. If you were that tiny, you would be inside of a cell. So that's what he's talking about here, is this constant back and forth between the mitochondria, of which you can have thousands, such as in the cardiac muscles, because depending on the energy requirements of that cell. And Frank has done incredible research into that area of being able to test mitochondrial function and be able to influence the production of energy. Pedersen has shown that the transition to anaerobic metabolism, it dramatically alters the landscape of the cell because, well, I'll get to Otto Warburg in a bit, but by binding a substance called hexokinase 2 to the mitochondrial outer membrane, it turns cells into immortal, crazed fermenters. Normally we have what's called aerobic metabolism in our cells, that being combining oxygen with sugar in the process of oxidative phosphorylation takes place in the mitochondria. And that gets shut down in cancer cells, and they move over to anaerobic metabolism or glycolysis. Now, Seyfried has shown that how damaged mitochondria send a distress signal, or what's called a retrograde response to the nucleus. And even in the presence of adequate oxygen, after it passes a certain critical point, which we haven't really determined yet, the transition into anaerobiosis becomes irreversible. Even in the presence of totally bathing that environment in oxygen, the cell will persist in using anaerobic metabolism. This signal tells the nucleus to transcribe a host of genes, responsible for preparing the cell to ferment glucose instead of aerobically burning it, in order to compensate for the declining oxidative energy production. And so in some respects, and there are some people that talk about, it's almost like a survival mechanism in some ways, that a cell will determine that its normal energy production can no longer function properly, and it switches over to anaerobic metabolism in order to try to survive. But obviously there are other consequences. And so then as this retrograde response becomes chronic and genomic signals transition the cell into a different cellular architecture, the legions of proteins designed to protect and repair the DNA begin to stand down, and those genes get turned off. And that's when you start to develop the DNA abnormalities. In 1948, Darlington noticed this phenomenon and wrote, "The development of unbalanced nuclei in tumors is without precedent in any living tissue. It implies a relaxation of detailed control of the nucleus, which is a constant process. That gets turned off." And this in turn argues that the nucleus is not itself directly responsible for what is going on. I remember when I learned this idea, and it was earth-moving to realize that this is actually the process that takes place. So we have the project called the Cancer Genome Atlas right there that was started in 2006, TCGA, to find the genes responsible for cancer because of the somatic mutation theory of disease. And 2006 was it wasn't when we started being able to sequence DNA, but it's when the process started becoming cheaper and cheaper. Remember when the human biome and the sequencing every gene in human genetics was the goal, and it took years to accomplish. And now you can send off a sample and get a report back in a few weeks. So this TCGA was a spectacular failure. They have not found the gene responsible for growing cancer. They developed over 700 drugs to try to address genetic abnormalities in the quest to find the, quote, cure for cancer. All of them failed except for one, Gleevec. Those of you familiar with that? And it exerts its effect by altering pathways turned on by damaged metabolism. So even though it led to the development of Gleevec, it supports the metabolic theory of the disease. And so now on to some striking evidence that took place back in the 1980s, over 30 years ago. There were two groups working independently, one in Vermont and one in Texas. And they performed a meticulous series of nuclear transfer experiments with shocking results. Warren Schaefer's group at the University of Vermont took the nucleus out of a cancer cell and transferred it into a healthy cell with its nucleus removed. The new cell contained the DNA of the cancer cell with all of its supposed driver mutations, but it had the cytoplasm and mitochondria of a non-cancerous cell. The Vermont group found that when they transplanted the new cells into 68 mice, only a single one grew a tumor over the course of a year. The cells containing mutations thought to be responsible for the disease were silenced by the healthy cytoplasm and mitochondria. Meanwhile, down in the University of Texas, Jerry Shea was doing similar research. He ran the same experiment, and not a single mouse developed a tumor, confirming the Vermont group research. In Texas, however, they went one more step, and what they did was they ran the experiment in reverse. They implanted the nucleus of a normal cell into a cancerous cell. If mutations to DNA cause cancer, the new cells should be normal, healthy cells. So what happened? The damage to the mitochondria, followed by a retrograde response to the nucleus, caused the new cells to become cancerous. That's pretty big food for thought as far as proving a metabolic theory of disease. When they transplanted the new cells containing a malignant cytoplasm and a normal nucleus into newborn mice, 97% of the mice developed tumors. This was in the 1980s. How many of you have heard about that research since then? Over 30 years ago. Even further back, we have the biochemical principles discovered and developed by German physician scientist Otto Warburg. Many of you have heard of him and realize that he received a Nobel Prize in medicine in 1931. Now, what's interesting about Otto Warburg is he was Jewish, and he stayed in Germany and was allowed, if you will, by Hitler because he was such an outstanding scientist. And he received the Nobel Prize in 1931, but Hitler, in his heyday, refused to allow any German scientists to receive a Nobel Prize during his reign. And Otto Warburg was nominated again during that time, but wasn't allowed to leave Germany to go accept his Nobel Prize. He proved in the 1920s that cancer cells of all types, that's where the concept of switching over to the anaerobic glycolysis metabolism of the cancer cells came from. It was Otto Warburg's research. Burning sugar without using oxygen. According to Otto Warburg, there is one and only one cause of cancer, and he maintained throughout his life that that cause was an inadequate supply of oxygen for cellular metabolism. Triggers the cell, for whatever reason, triggers the cell to switch over to anaerobic metabolism. So I tell my patients, and I can tell you that 99.9% of people that come in with a diagnosis of cancer have heard somewhere, cancer loves sugar. And I tell them, cancer doesn't love sugar. Cancer requires 18 times more sugar than normal cells because anaerobic metabolism is 18 times less efficient at turning a given amount of sugar into a given amount of energy. And so cancer does crave sugar, but not because it loves it, because it can't survive without it. But at the same time, I explain to them, you cannot cure your cancer by not eating sugar, because 85 to 90% of those people that have heard that concept, they eagerly tell me, I stopped eating sugar. And they feel like they're killing their cancer. And I tell them, it's impossible to do that because your brain requires sugar. And if you drop your blood sugar down to zero, which you can't really do on your own, but we could with a dose of IV insulin, you will die because your brain requires that sugar at a certain level. However, there's a way to deal with it, and I'll get to that. Okay, so when this change to anaerobic metabolism happens, two major shifts take place in the mitochondria, which, as I mentioned, are the energy furnaces of the cell. First, oxidative phosphorylation is shut down. Second, the apoptosis program or the self-suicide program that's contained in every single one of our cells is also shut down. And this is what allows cancer to grow out of control. Your normal cells have a program that gets triggered if there is physical or chemical damage beyond repair, or if they hit the age wall, whatever that may be. The apoptosis program is triggered and the cell commits suicide. And in cancer, that process gets shut off. And that's why cancer can grow out of control. So there's a picture of the cell and all the organelles contained in it and all the stuff that's happening. We tend to forget about that because we're not conscious of what's going on inside of our cells. But it's an unbelievably complex process. Okay, what can we do about it? And that's what it comes down to, because theories are theories, but if you can't turn it into practice, then what use is a theory? So the short answer to that question is ketosis, metabolic therapies, and free radical generation, and apoptosis enhancement. The long answer is going to take a little bit of time, but I will be able to cover it. So as I mentioned a little bit ago, you cannot just stop eating sugar because the brain needs sugar. However, a key principle to understand is that all of our normal cells, and most especially the brain cells, can switch over and burn ketones for energy if there is inadequate sugar supply. An interesting concept is that, or not a concept, a fact, is that if the cells in the body are given a choice between ketones and sugar, they will always choose sugar. It's like your 5-year-old. The sugar is the primary focus. But if you create a situation where there's inadequate sugar levels and adequate ketone levels, the cells can easily switch over and start metabolizing the ketones. Now, where do ketones come from? The liver makes ketones when it burns fat. Ketones themselves have been shown to have somewhat of an anti-cancer effect on their own, but we've all heard of the ketogenic diet, and certainly most of us have heard of the Atkins Phase 1 diet, which is a ketogenic diet. But at the same time, in our medical training, we've learned about diabetic ketoacidosis, the big bad monster, because in fact, it can kill people. So it is a very dangerous condition. But I am not talking about diabetic ketoacidosis. I'm talking about metabolic ketosis, and they are different as night and day. Dr. Russell Wilder at the Mayo Clinic in 1924, he developed a highly effective therapy for treating intractable epilepsy. And of course, in 1924, we didn't have the anti-seizure drugs that we have today. And he developed a very effective therapy by putting people on a ketogenic diet, generating ketones, keeping the sugar low, and lo and behold, the seizures stopped. Well, it fell out of favor in the 1940s because of the development of anti-seizure medications, which have a time and a place. However, what happened in 1994, a young boy named Charlie Abraham, his parents started the Charlie Foundation after he completely recovered from daily seizures, despite being on all the full array of anti-seizure medications. And his parents uncovered this information about the medical past and decided to try it, and it worked. He started on the diet as a toddler. He remained on the diet for 5 years because I regularly get questioned about, is a ketogenic diet safe? The answer is unequivocally yes, it is. He's now a college student and remains seizure-free. So again, we're talking about metabolic ketosis versus diabetic ketoacidosis. Now, in regards to seizures, it's an interesting thing that Hippocrates, theoretically the father of modern medicine, even though nutrition was a hallmark of his therapy because they didn't have drugs at the time, but they didn't call it epilepsy or seizures, they called it fits, having fits. And one of the most effective things they found to stop the fits in people that had them was to have them fast. And one of the quickest way to put someone in ketosis is to have them fast, go on a water fast. And they will get into ketosis very fast. So the father of modern medicine was using ketogenic diet as a therapy, and it wasn't called that then, so he didn't even know that. So I have, I wrote a whole article, which I didn't bring up with me, but if you're interested, we can talk after this lecture about, is a ketogenic diet safe? And as I said earlier, unequivocally, yes, the answer is yes. And in some situations, bodybuilders and athletes have found their performance increase and improve when they're in ketosis. Ketones are like high-octane gas in a Ferrari. And many people find that their thinking is sharper and their energy levels are higher. Because you would think, right offhand, you would think if you fast, you stop taking in fuel, that you'll get tired and you'll be hungry. And that is true initially, but if you've ever tried a fast, a water fast, after the first roughly 48 hours, you don't get hungry anymore. And there is a surge in energy. Now, of course, that's self-limiting, but just interesting to note because it makes me think, what's happening inside my body that I'm feeling okay with not eating? Because we tend to think that if you don't eat, you're going to drop over from exhaustion. So given the fact that this ketosis is safe, then how can we use that in the fight against cancer? And the concept to get across here is that free radical generation is a route to killing cancer cells without harming normal cells. And as homeopaths in the state of Nevada, we have at our disposal the whole category of what is called oxidative therapies: ozone, hydrogen peroxide, high-dose vitamin C, the ultraviolet light treatments, and even up to and including IPT with the older, far less expensive chemo drugs. They knew 50 years ago that free radicals killed cancer cells, and the first chemos were designed or chosen because they generated free radicals. But when you go to full dose chemo doses and totally blast the body with the free radicals can be very damaging. But the metabolic facts are that aerobic metabolism, using oxygen to burn the sugar, one of the byproducts is free radicals. Free radicals are free oxygen molecules, and they are neutralized by what's called antioxidants. I'm sure everyone in this room has heard that term before. And we have some internal antioxidant metabolic pathways in our normal cells because they've been forced to develop those because they're challenged by the free radical generation. Anaerobic glycolysis does not produce free radicals. So cancer cells have never been challenged to develop these internal pathways to be able to utilize antioxidants to protect themselves. So the whole concept is to, okay, I tend to forget about what the slides are doing. Okay, so some of these antioxidant pathways are glutathione, superoxide dismutase. I'm sure there's even ones we don't know, we haven't discovered yet that are at work. because on a practical level, the cells either developed them or they died. And so they developed them. But the cancer cells never did because they haven't been challenged to do that. So in to me, again, earth-moving research study done by Thomas Seyfried and another PhD named D'Agostino in Florida, they took a population of mice with glioblastomas. Now, those of you that understand cancer realize that glioblastomas are one of the most aggressive brain tumors, and most regular cancer therapies do not work against them. And basically, the major therapy that's utilized is let's blast the whole brain with radiation and hope that we kill all the cancer cells. And yeah, there's always collateral damage with radiation. But I've told my patients, if you picture yourself in a nursing home drooling in your lap, but the cancer's gone from your brain, realize that you'll be in the record book as a successful treatment. And most people decline that future for themselves. In any case, what Seyfried and D'Agostino did is they put these mice on a ketogenic diet, pushed them into ketosis, and of course they had total control of their intake because they're lab mice. And so they put them into ketosis and they put them through hyperbaric oxygen treatments. Now, on the surface, you'd look at that, if you know anything about hyperbaric oxygen, that they're saturating the tissues with oxygen. But I already told you that research has shown that cancer cells, even in the presence of a saturated oxygen environment, do not succumb to it, certainly, and they do not switch back over to aerobic metabolism. They continue to carry on their glycolysis process. However, saturating the tissue with oxygen, I also just told you, free radicals are free oxygen molecules. And hyperbaric treatments generate massive quantities of free radicals. And what they found, and if you want to see the study, I brought it with me. I got it off of PubMed. They had almost 80% of the mice, the tumors shrank up, and their survival time was massively longer than the control group. And so when I first read that study and heard about it in Tripping Over the Truth, that book Mike told me about, I was struck by the fact that we have at our disposal a whole category of therapies that are poo-pooed, laughed at, or totally suppressed in most other states, that being the oxidative therapies. And how many times have we had Frank talk about ozone? And we've been, many of us have been using IV peroxide and high-dose vitamin C. And if you don't understand vitamin C and think, oh my God, ascorbic acid is an antioxidant, that is true, but above 10 grams a day intravenously, it stimulates the polymorphonucleoside white cells to produce hydrogen peroxide internally. That's been proven. And it becomes an oxidative therapy at high doses. The ultraviolet treatments we have, UBI treatments and UVLRX is the new kid on the block, which is actually a probe that fits intravenously and radiates ultraviolet light right into your circulation. And then IPT with the chemos, as I mentioned. All of these produce what I call a free radical storm. And if you can get the patient into ketosis, and we in our office developed a branch off of the IPT tree. I've been doing IPT since 2004. Dr. Brody sent me down to Tijuana to study with Donato Perez Garcia, the grandson of the guy that developed IPT back in the 1920s. We do we call KPT, ketogenic potentiated therapy. And we get our patients into ketosis. There's new meters on the market today, you can buy them on Amazon, that do a finger poke and give you sugar and ketone levels, blood ketone levels, and that's more accurate than urine testing. And when we can get people into certain ranges, and we don't take them down to the ultra low blood sugar levels like we used to, because to bring them out of that trough, you have to give them dextrose, IV dextrose. And one of the quickest ways to kick someone out of ketosis, I told you, if the body has a choice between sugar and ketones, it will choose the sugar. And you will be out of ketosis pretty quickly. So I was challenged by my nurses saying, how are you going to do IPT if 2 times a week you're kicking people out of ketosis? And they've started calling me ketogenic Bob, because I'm ketogenic all the time. And so we sat down and talked about how if we can get people into ketosis and have their blood sugar levels be down in the 60s, which is still in the normal range, but the far lower edge of it. And then when we're finished with the treatment, give them protein and fat to bring them back out of the it's not really hypoglycemia, but physiological hypoglycemia, and back into a higher level of blood sugar. And we are seeing some really, really good results. And so that's the research by Seyfried and D'Agostino, I believe, was pivotal in starting to show the possibilities here for expanding this kind of therapy. And we find ourselves, I believe, our profession in this state, in a very unique situation, because I talk to other doctors from other states all the time, and they're either afraid or they have actually been harassed by their medical boards over doing some of these therapies. And oxidative therapies is one of the cornerstones of our profession in this state. So there we have the what I mentioned as the oxidative therapies. And our goal is generating free radicals. Now, at the same time, we are utilizing what I call metabolic therapies designed to affect different aspects of the metabolism of the cell. Salicinium we refer to as a fake sugar. It's a glycobenzaldehyde. It's a glucose molecule with a benzaldehyde ring. And there's an enzyme that's only contained in cancer cells. It's the same one that splits the cyanide off of the laetrile, beta-glucosidase. Splits off the glucose molecule and frees up a benzaldehyde ring. Benzaldehyde is a highly cellular toxic chemical, and in very low concentrations will kill the cancer cells, but it's only contained internally in the cancer cell. Poly-MBA affects the metabolism of the mitochondria. We're hoping to be able to turn those mitochondria back on again long enough to affect that apoptosis program, and that's specifically what DCA is designed to do. It was proven in studies at the University of Alberta, up to and including humans, not just mice, to be able to activate the inactivated mitochondria long enough for them to trigger the apoptosis program in the cancer cells. And so the poly-MVA is designed to support that energy production in the mitochondria to get them to stay alive long enough to figure out that they're extremely abnormal and they don't belong there and they should leave. So I like to think of the whole process to relocate these cancer cells instead of kill, because the war analogies trigger, even on a subconscious level, trigger the classic parasympathetic nervous system, the sympathetic process, which is fight, flight, or fright. There is no more a sympathetic dominant human activity on this planet than warfare. And warfare, we know that sympathetic overstimulation suppresses the immune system, shuts off what's considered by the subconscious mind unessential processes in the body so that all energy can be focused on survival, muscles, heart, breathing. brain reflexes. It's a totally subconscious process. So if you stimulate that by saying, kill your cancer, we're going to fight cancer, we're going to picture different battle scenarios, you are triggering sympathetic overdrive and hampering the healing in that patient. 3BP, bromopyruvate. Those of you that know about Dr. Koh's research at Johns Hopkins, it's a little bit of a dicey situation because we can talk about it after the lecture. Any one of these topics could be food for one lecture, but the 3BP simulates lactate and goes back into the cancer cell. Cancer cells can burn lactate for energy, and the 3BP they cannot. And it simulates the lactate, goes in, shuts down energy production in the cytoplasm. And when that happens on a cellular level, cells die. Butrex is the salt of butyric acid. Both Butrex and DCA have research history of being able to re-stimulate apoptosis. So that's why I referred to them as metabolic therapies. And then KPT, I mentioned. IPT, for those of you that don't know, I assume most of you do, was developed in Mexico in the 1920s. You basically lower the blood sugar with IV insulin, and at a certain point, referred to as the therapeutic window, you give a number of different substances combined with a tiny dose of sugar. And just quickly, we use Laetrile or B17, salicinium, poly-MVA, and the chemo drugs, but only at 10% of the full dose. So we get 90% less of the, quote, bad effects. I don't believe there's any such thing as a side effect. There are only direct effects. If you give a drug and you get an effect, that's a direct effect by definition. Side effects are things that doctors don't want to have happen, or they don't know they're going to happen until they happen. And then they get labeled as side effects, and they're either stuck in a cubbyhole, that's just a side effect, or I can write you a new prescription to deal with that side effect. And so I prefer to call them all direct effects, and they're good or bad effects. And so there is a place for chemo, and I explain to patients when they hear that word, a lot of them cringe because of history, and they know what happened to Uncle Charlie with his prostate cancer, and they don't want to see themselves turn into that. And but when I explain to them these concepts that I just talked to you about, which I do to all my patients, and the fact that we're using 90% less of a dose, that there's 90% less of the bad effects as well. But at the same time, we're targeting those cancer cells in a very specific way because we're able to utilize that anaerobic metabolism and the lower efficiency to our advantage. So the goal of this lecture was to get you to consider that cancer is a metabolic disease. With that as a given, diet and less toxic proven therapies can be chosen to address the cancer at a more fundamental level than has been done in the past. And then I have an unabashed plug for my new book, Outmaneuver Cancer, and it does not teach you how to do these therapies. It goes over the things that I've just covered. And if you're interested in learning this stuff, give me a call. I tell my nurses I'm more excited today than on the day I graduated from medical school, which was in 1977. Are there any questions? Well, it means one of two things. Either it totally went over your head, or I answered all of them already.
Hello. Thank you, Dr. Bob. Do we have any questions? Great stuff. Congratulations.